Done, sir. Done, sir.
Today, we are very happy to have an eminent professor and scientist, Dr. Seema Madhuria, with us. She will speak to us on the damage to the stones of monuments, etc., and how bacteria can help in sealing micro cracks in the stone monuments. So I request Madam Babita Sharma to please introduce. Babita, we cannot hear you.
Should I, sir? Should
Yes,
I? Yeah,
please.
yes. Okay, sir. Good evening, everyone. My name is Babita Sharma, and on behalf of Microbiologist Society of India, Chandigarh, I'm here to welcome Dr. Seema Bhadoria, who is our today's guest speaker, and she'll deliver her talk on biodeterioration of stones and their bioremediation. Before proceeding further, I would like to welcome Dr. A.M. Deshmukh, former professor and head of Department of Microbiology, and Dr. Baba Saheb Ambedkar, Marathwada University, Aurangabad. He's also president of Microbiologist Society. We welcome you, sir. Sir is here with us, sir. Oh, okay. I would also like to acknowledge the efforts of Dr. Swaranjit Singh, who took the initiative to organize such informative talks and other activities. I would like to introduce Dr. Swaranjit Singh to our speaker and the audience. Dr. Swaranjit Singh is an eminent scientist, and his name has been listed in top 2% global scientists for consecutive 3 years. He's the first IDA in India and has served in MTCC and Gene Bank. He's an eminent member in 2 WFCC committees. He's the member of Task Force National Biodiversity Authority, Chennai. He has been awarded Loyola Environmental Award. He was the former head Environmental Biotechnology and Microbial Biochemistry, CSIR IM Tech. He was also facilitated with fellow National Environment Science Award. He also got Lifetime Achievement Award in 2022 by St. Xavier College, Ranchi, and Lifetime Achievement Award in 2023 by Shulini University, Solan, HP. His publications are in high impact factor journals, and the articles are highly cited in their literature. With H index 58, I-20 index 150, and cited 18200 times. We welcome you, sir. I take the honor to welcome Dr. Seema Bhadoria, Principal of Balwant Vidyapeeth Rural Institute, Bichpuri, a government-aided college at Agra. She has also served as a microbiologist and professor and ex-head of botany department of Raja Balwant Singh College, Agra. Her field is bioremediation of polluted environment. She is founder of RBS Community College for Extension of Organic Farming, and she has contributed in 14 innovative products. Few of them are vermicompost from PETA and agriculture waste, biofertilizers and biopesticides, rapid seed testing kits, biodetergents, bioremediator pack for a mole of bag deposit from all the stone surfaces, nano emulsions for stone and artifacts, Panchagavya, nano herbal bandage. These are innovative products which ma'am has contributed. She has synthesized bioplastic from cornstarch and agro-waste starch granules. 5 patents, 15 products, 4 technology transferred to companies, and 50 PhD awarded, along with 14 books she has published, 175 papers she has published. She is having 7 international research collaborations with various international universities and corporates in Russia, Japan, Italy, Mexico, United Kingdom, Israel, Indonesia, under International Exchange Program of Department of Science and Technology in New Delhi. She is honored with most prestigious National Award of Women's Development through application of Science of Technology by the Ministry of Science and Technology, Government of India in 2011. She had also been graced by the Devi Award presented by the Chief Minister of UP in 2019. She had been IQAC coordinator as well as NAC coordinator and research and development coordinator under DSIR. We welcome you, ma'am. I hope I am audible. I hope I am audible.
Yes, yes, you are audible.
Yes, yes.
Okay. We welcome you, ma'am.
Thank you. Thank you. Thank you very much, Dr. Babita. It's a great pleasure to be here on the forum provided by Professor Swaranjit sir. It's a real opportunity that what we have been working in the field of microbiology since last 25 years may be put before the Microbiologist Association to show that this is also a field in which we can contribute our dedication, and also with the help of microorganisms, we can do something better for the society and our cultural heritage. On this occasion, which is really an appreciable moment for me, and I'm really enthusiastic to show that when I was working in Department of Botany in my lab, Microbiology and Nanotechnology Research Lab, I came to a problem that was an identification of microorganisms. Therefore, I met Professor Swaranjit sir when he was in IMT, and he supported me with lot of help and also identified the microorganisms till gene levels. So that was at least 10 to 15 years before. And till now, we are contributing with his support for in this field. So I thank, sir, this is on this forum, I can thank your contribution for my development. Today, I shall be talking on an aspect which is very important because buildings, constructions, hotels, institutions, heritage, monuments, caves, forts, everything, they are being constructed with the help of stone or cement or concrete or all the building materials which are required for developing a building. But we face that these buildings are being developed in eras and we want to maintain them for long period for our future generation to show that our historical remnants which are on the earth can be shown to them. They should be attached to all these things. So this topic actually encouraged me to understand what is the mechanism of the biodeterioration and how microorganisms actually attack on these substrates and how we can prevent them with the same microorganisms which can be useful for their sustenance. So first we shall understand what is the biodeterioration of the stone. Although every microbiologist understands that biodeterioration and biodegradation are two concepts, In biodeterioration, the economic wealth and economics and also the we can say the value of a substance degrades. While in case of biodegradation, we can have any valuable product which can be developed and can be more of better having better economics. So in case of biodeterioration, the stone is a substance which is made up of many things. The constituents are chemically very strong, but they are prone to the attack of the microorganisms through the acids which are produced by them. So the stone cultural heritage materials are at risk of biodeterioration, which is caused by a diverse population of microorganisms which are living in the biofilms. Secondly, the microbial metabolites which are produced by the biofilms are responsible for the deterioration of the underlying substratum and may lead to physical weakening and discoloring of the stone. The first step by which we can understand that stone is undergoing biodeterioration is physical observation. We can see the change in the color, or we can also see that there are porous structures which are being formed, or there can be other things which have taken place due to the intervention done by the microorganisms. The fundamental process for biodeterioration of stone involves microbially mediated reactions, that is, oxidation, reduction, acid dissolution, and elemental cycling, which results from the activities of the environmental microorganisms. In stone, we can say that phototrophic organisms, especially cyanobacteria, are the first participants, which are being followed by archaea and also fungi. The microorganisms and biofilms can penetrate and create fissures or cracks in stone. Here, I would like to say that when the microbial mediated reactions are taking place on the stone surface, the dissolution takes place with the help of the acids. The chemistry behind the stone deterioration is the stone consists of granules of different minerals and rocks which are glued together with clay, lime, and other substances. We can see that there are main oxides of silica oxide, aluminum oxide, calcium oxide, iron oxide, etc. And these are all specifically of metal based. Then stone deterioration results from both free radical reactions and ionic reactions. Free radicals are formed due to homolysis that are electrically neutral and carry single unpaired electron. That is why a radical chain auto-oxidation reaction takes place, in which I is the initiator, it propagates, and then chain branching takes place, and finally the termination with the acids which are the end products. These acids actually take the chain reaction, hold true for the rates of the reaction in oil and varnish films of various kinds applied on the stone surface. Then these varnishes and oils, they are used to make the surface smooth. That is why they act as an initiator for the chain reaction. Light at particular wavelength and other atmospheric agents of deterioration bring these changes and make the surface rough. So the first step is discoloration, and second, it becomes porous. What problems we are facing? In this picture, this is a Kailash temple. Nowadays in Agra, when Shiv Savan is going on, this temple is highly pressurized demographically. All the people, they are going for worship, but the condition of the temple is very clear. Here you can see that there are grasses. These are the stone inhabiting vegetation which is growing on the surface, and the blackening of the areas, and here the crust is formed. Similarly, here the black crust is formed, and this is the condition. We can see that the sandstone is inhabited by the microflora. This is Orcha temple, which is known as Ram temple in Orcha. Here also we can see the exfoliation of the upper layer of the sandstone is taking place, along with the blackening and deposition of soots over the building. Here we can see that with the left of the excreta of pigeon has made the whole surface black while the building is white. This is actually Meloti temple, which is 108 years old, and it was made up of terracotta. Here you can see that the vegetation is growing on the top of the temple, and the roots have penetrated inside it, and just they have destroyed the heritage value of the temple. This is Northeast temple. Here also we can see the high humidity has enforced the microbial population to exist on it. This is a temple, Shiv temple, and here also we can see that seepage of rainwater in the cracks, and also this poor drainage and alkaline surface, they have also actually deteriorated the condition of the temple. This is another temple which we can see. This is Khajuraho. Here you can see that the black soot deposition is there. This is Yaman temple. This is Lakshman temple, Ghantai temple. Each and every temple we can see that sculptures, statues, they are all facing the microbial attack. These are churches. This is the Shimla. This church is in Shimla. Everyone must have seen it. Because of regular maintenance, it is in good condition. But the oldest church, the condition is, we can see here. And this is St. George's Church in Hyderabad. So this is Ellora, and Dr. Deshmukh must have visited these places. I think Ellora, Ajanta caves are very near to Aurangabad, and there this condition is existing, and they are very old temples, very old caves, which are being attacked by the cyanobacteria, and blue-green algae, the archaea also are existing. These are Ajanta caves. The paintings are being attacked by the microorganisms. Ajanta caves, you can see here. This is also one of the portion from the caves. This is a cemetery on which we have performed our experiment. Coming to the focus that what we have done and what we expect from the microbiologist in case of maintaining our cultural heritage. This cemetery is more than 100 years old, and this was facing the white deposition of the alkaline salts. Also, here we can see the blackening of the surface, here the blackening, and here the soot deposition is there. So we go to the site, observe the deterioration patches, and then we collect samples for isolations. This is Amer Fort from Jaipur. The conditions is there. This is also Amer Fort. This is Agra Fort. The condition you can see, the same problem is there. And this is Leela Gumbaj Ki Masjid. So we can classify the type, the deterioration type of the stone, like first black patina and exfoliation. Exfoliation In case of exfoliation, the layers are separated. The sandstone is dissolved, and what happens? The upper layer, which is very sheen and very clean, that layer is separated, and there is formation of black patinas. Second is exfoliation of stone. This is Fatehpur Sikri. Here the salt is deposited, salty water is there, due to which this exfoliation takes place. The layers are separated, just destroying the beautiful architecture which is present here. Very nice designs are made, but due to exfoliations, everything goes bad. Similarly, here, this is black soot deposition. This is first stage by which the deterioration starts. So patinas are of orange color, green color, black color. This is sulfatation. Here we can see deposition of sulfates. This is pitting. The holes are there. This is causing pitting. We can see these holes existing on it. And this is flaking, engrooving. The flakes are coming up. So these are more types by which we can define how the deterioration takes place and how we can see that deterioration is occurring. So we started our research project. This is actually a collaborated project with the Indo-Italian government and our Ministry of Science and Technology, in which we collected the stone samples which were deteriorated in the nearby the monuments. So here we can see that it is very clear the deposition of the microbes. Here the blue-green algae is there. So these are the deteriorated stone samples which we collected. Then we went for isolation. It is very simple how to isolate the microorganisms which are occurring on the stone and are playing role in deteriorating the surfaces. This is the first method, which is called as tape method of isolation. In this, we take a simple sterilized tape and go to the site. We place the tape on the affected area and just to scratch it. First we paste it on the surface and then scratch it. We have our sterilized petri dishes having the media, and then we place our strip on the media and incubate in the incubator. So here, this is actually the tape method of isolation for Kailash temple. And in this, you can see very clearly that microorganisms are occurring, are there on the plate. And especially this is Rose Bengal. So in Rose Bengal media, we go for fungi. So here, Trichoderma, Aspergillus, Carvularia, Alternaria, Fusarium, these are available, or also Stachybotrys. So these are the prominent microorganisms which are seen. Then another method for isolation is swabbing method. For this, we take a swab of High Media Company. Also, we can do another cheap method with the help of Johnson bud by sterilizing it. But we had selected high media swabs. These swabs we take to the site and then we just run it over the surface of the stone where the affected area is available. After taking this swab, we come to the laboratory and then we just run the swab over the culture media And the another plate is the control plate, in which there is without any inoculum, and then this is incubated, and we can see that black fungus are more dominating in the swab method. This is bipolaris, Carvularia is there, Alternaria is there, Stachybotrys is there, and few more. Epicocum is also there, and also Fusarium. So this is how the swabbing method of isolation is done. This is direct plate method of isolation, in which we take we just scratch the affected surface little bit with the permission of the Archaeological Survey of India, that we can take small pieces, and then we can then we will bring to the laboratory and culture them on our culture plate. So this is direct plate method, and then after isolation, culturing and isolation, then purification is done. So this is purification. In this we can see that Fusarium roseum, Phoma, Stachybotrys, there are some, I have shown few pictures, some microorganisms which are present, which we had isolated. After this is bacteria, Pseudomonas aerogenesa, Micrococcus roseus, Nocardia, Bacillus globiformis, Micrococcus luteus. So these are the bacteria which we had isolated. Many were isolated, but few are here. These are the photomicrographs of the microorganisms. Aspergillus fumigatus is there, Stachybotrys, Fusarium oxysporum, Aspergillus sedovi is there, Bipolaris is there, Microsporum is there. Microsporum is actually, we can say it is a fungi which is due to keratinophilic activity. And this is Rhizopus, alternata is there. We also found Elga, cyanobacteria available. So this is Eulothrix, Langibia, Myxosarcina is there. And this is the microbial biofilm, which was observed with the help of PAS staining. In this, the surface is completely covered. In the first picture, you can see that surface is completely covered with the microbial film. But in the B, the biofilm is reduced in the subsurface. And in the C, no biological biofilm is visible in the interior of the stone, which means that the upper surface is attacked by the biofilm. Substantially, the second layer subsurface is attacked, but the last layer, that is interior of the stone, is not attacked, but slowly the deterioration takes place. This is electron microscopy of the lichen thallus is growing on the surface of the stone, and there is fungal contamination here. The fungus, you can see the mycelium growing on the stone. So it is penetrating inside the stone surface. So this is electron microscopy, which we had done. These are the bacterial cells which are adhering to the stone surface. It is very clear. We can see these cells here. And this is biopitting. The infested stone, this is fungal mycelium, and this is the biopitting and dissolution. The stone has dissolved completely. And here we can see that mycelium has covered it. This is the growth of the yeast on the stone surface. This is very clear. This is erect fungal mycelia. Just these are the fungal mycelia showing exopolymeric substances, EPS. This is the stone in which the algal filaments have penetrated and the stone is softened. Just see, it is broken, just like osteoporosis in our body. So the stone has dissolved. This is the close-up view that how the penetration takes place of the algal filament and the stone particles are breaking up. Finally, we then go for understanding what are the substances which are available on the stone surface which are attracting the microbial growth. So they may be pollutants, they may be oil, they may be interferences which are being done by the humans and keratins. So we go for XRD analysis of the stone, and we found that there is presence of benzoquinone, which is a pollutant, or it is a fungal melanin, which is produced by the microbial action of on the stone surface. So XRD, electron microscopy, and normal microscopy and isolation methods, microbial methods, these are the techniques which are to be used for studying the biodeterioration process. Then comes, this is the problem, but how to restore it? For restoration, what happened during this project, we came to know that there is a Japanese paper which is used for conservation and restoration of the stone surfaces, which is very expensive. And in Italy also, in Milan and Florence, I saw that they were using this Japanese paper for applying gel impregnated with the bacterial cells. So what we thought that this gel is expensive, this paper is expensive. Can we replace it with the multani mitti, which is already been used for cleaning these surfaces? So we started our restoration process with this. Here you can see that we have selected the same symmetry which I showed you before. In this symmetry, the surface is black, you can see, and we impregnated Pseudomonas aeruginosa. This was actually completely identified and confirmed with the help of Dr. Swaranjit Singh from MTech. And also we have gone for its, we have submitted this strain to MTech. We impregnated this Pseudomonas aeruginosa cells in Multani mitti, and we made it like a poultice, and we applied it over the surface and covered with the polythene, a plastic film. So here we can see very easily that after the removal, after removing this surface, after 24 hours, the bacterial growth was there and the surface was comparatively clean. Similarly here, this is the area we compared other bacteria also, because we identified many bioremediators and we wanted to find out bioremediators from the same stuff. So we identified this Bacillus, we have taken Bacillus subtilis also and Pseudomonas aeruginosa, and we compared their activity. Here we can see again Pseudomonas aeruginosa is giving nice, making it more clear in comparison to Bacillus subtilis. Here we have applied Bacillus globiformis, and we can see that it has not shown very good results. This is carbogel. This is actually the Japanese method. This is the picture of the surface of Italian symmetry. There is a flower here, and they have, this is the test microorganisms, and this one is the control one. This is Bacillus, subtilis, and this is Pseudomonas. After its removal, Pseudomonas aeruginosa gave a better result, and Bacillus subtilis could not give very nice results. So comparatively, this is more better. This is laboratory practical, which we conducted in Florence. In this, we have taken marble instead of sandstone. And here, the sandstone pieces we had taken from Gokulpura in Agra and some from Swami Bagh. And then we have planned this experiment in under different humidity, under different temperatures and controlled conditions to see whether in laboratory conditions also the black crust is appearing or not. So we found that after 216 hours, a black crust was developed on the top layer of the marble. So it exists in the environment, and it can be restored with the help of the microorganisms. Here also, we have developed a soft gel with the help of nanomaterials, and these soft gels are being applied to the surfaces, and then we found that these methods can be used for removing the patina and the different deposits which are on our cultural heritage objects. This is black crust removal from synthetically generated gypsum layer. So instead of multani mitti, we also tried gypsum, and we found that here with the help of electron microscopy, we can see that this is also giving a very nice results, and this crust can be, this gypsum layer can be synthesized in the laboratory and can be also used for bacterial treatment. How we can confirm that bacteria is living or non-living in the poultice which we have made? So here we can see in the electron microscope the sample. This was done in Jalma Institute, where we have TEM and SAM. So here in the first picture, it is clear that there are no bacterial cells, but here which we have impregnated the bacteria, they are seen and they are existing. So we have to confirm that Multani mitti is supporting its growth or not. Otherwise, just putting the microorganism and thinking that it will do the work, it will not. So we have to see that after removal, these bacteria do not have any side effects also. So here we can see in the carbogel also, we compared the results. Here we are able to see the bacteria, and here this is without any bacteria. So this is the symmetry of Florence, where we had worked for it. This is marble based. So different types of stones we have worked upon. This is again of Milan. This is deteriorated surface, and this was cleaned by the team of Italian government. So that's all which we have done, and much more. For this Multani mitti package, which we have transferred to Archaeological Survey of India also, we have achieved an international patent for this. So thank you very much.
During the rough breaking phase,
Thank you very much, Professor Seema, for the wonderful talk you gave.
Thank you, sir.
We really enjoyed seeing your beautiful PPTs, which were so clear, so vivid. And we know many things now about, damage to stones and the effect of microbes. Thank you very much for the nice talk. Now, Chandrayaan is about
Thank you,
to
sir, for
land
giving
on the moon.
opportunity.
Yes, sir. Thank you, Sima ma'am. I would like to thank Dr. Sarabjit also for planning and organizing such informative talks by eminent dignitaries whose experience tends to motivate all of us and also make us aware about such significant concerns. Thank you, sir. I am also thankful to Dr. A.M. Deshmukh for sparing time for us and to be the part of this discussion. Thank you, sir. Last but not the least, I am also thankful to the audience for participating in today's event.
Thank you very much, Babita Sharma,
Thank you,
for
sir.
the nice introduction, number one. And number two, a beautiful vote of thanks. Now I want to see Madam Seema Baduria. I could not see you.
Okay, sir, just a minute.
Yes.
Let's open the camera.
Please. Please.
Okay.
It does not open.
Yes, sir. Just I'm just seeing.
You know, the Chandrayaan is about to land very soon.
Yeah.
And they'll collect the stones from the moon. And we'll give you some. We'll give you some of those stones also.
I'll stop sharing the screen. Then, so you have to allow my video to open.
It's all right. Some other time we'll do it. I'll do a video call with you some other time.
Okay, sir.
And thank you so much again.
Camera call.
Thanks to everybody. All right, Babita, then I think we can conclude.
Yes, sir.
For
yes,
today. And let us see the Chandrayaan 3 now.
Yes, sir. Absolutely, sir.
Okay,
Thank you.
madam.
Okay, sir.
See you again. See you.